particular cell type. We then take that data and we can ask the question about what is the relationship of genetic regulation of gene expression in this region of the genome to disease susceptibility using a statistical approach called co-localization. And what that does is it asks the question whether the genomic region regulating this gene in this particular cell type directly overlaps with the same genetic region of the genome that confers risk to a particular disease. So this is data from our lab, where on the top here we have the EQTL Manhattan plot, and we can see that the lead SNP here, associated with the expression of PCAM gene in microglia, is exactly the same as the lead SNP, which confers risk to Alzheimer's disease at genome-wide significance at this particular locus. And so intuitively, this alignment or co-localization would implicate the genetic regulation of PCAM in microglia with susceptibility to Alzheimer's disease. So it's clear that MR and co-localization are two approaches to inferring the causal relationships between exposures and phenotypes, and they have similarities but also important differences that make their combined use synergistic in terms of undertaking these types of analyses. So I've taken this slide from Verina's review of the differences and uses of MR and co-localization published in the American Journal of Human Genetics a couple of years ago. So with MR, the output is a test statistic and a p-value, but with co-localization, it's a posterior probability of between 0 and 1. MR, as we'll see, tests only one or a few genetic variants at a particular genetic region following careful selection of the instrumental variables, whereas co-localization requires dense coverage of SNPs across the genomic regions. Importantly, though, MR will provide us with information on the direction and the effect size of the relationship between the exposure and the outcome. And so that information gives us the therapeutic strategy that we need to adopt to mitigate disease risk, whereas co-localization doesn't provide this information. And as we heard from Verina, there's a higher probability of false positives with MR, since even weak associations between a variant and an outcome can yield a positive MR association. Whereas with co-localization, the statistics are more conservative because of the use of skeptical priors, and in fact requires strong evidence of co-localization to conclude that there is co-localization. So this is work that we have currently under review with Nature Genetics, which is work that we've recently completed where we combine co-localization and MR using single-cell sequencing data from nearly 400 brains, consisting of 183 controls. And these were the brains of people who had died without a neurological diagnosis and whose brains were neuropathologically normal on inspection, and 208 subjects who had died with a neurological diagnosis, mainly either Alzheimer's disease, Parkinson's, or multiple sclerosis. We generated single-cell sequencing data on these brains, which gave us nearly 2.5 million single-cell data. All the subjects were genotyped. And we integrated this with genetic risk data from GWAS studies for 41 diverse brain phenotypes. We calculated cell-type-specific EQTLs, as I've described. We also looked at the effects of disease on the genetic regulation of gene expression in the human brain, and I'll talk about that in a moment. We combined co-localization and MR to identify putatively causal associations between the gene expression levels in single cell types and disease risk. And then we looked at trying to replicate some of these using protein QTL data in both the brain and also plasma with data from UK Biobank. So this was our data set. As I mentioned, we had started with 409 genotyped adult donors, but we ended up with 391 post-quality control, roughly half controls and disease cases. And overall, we had gene expression on 2.35 million individual cells with about 4,500 cells per individual. And we annotated across these 8 major cell types of the human brain. So we calculated EQTLs in these individual 8 cell types using the approach I've just described. And across the full mixed disease case and control cohort, we found one or more regulatory variant for over 15,000 genes. And here on top of the total number of E genes for a particular cell type, and shown below that are the number of E genes that were specific for that specific cell type and not found in other cell types. Interestingly, what we found, and so this is a Venn diagram of the number of E genes that we found in the single cell data compared to an equivalently sized bulk tissue RNA sequencing data set, which we generated artificially by aggregating all the counts for each individual gene across the entire data set, so not taking into account the single cell type information. And what we found was that around 50% of E genes are only detectable using single cell data. And I think this shows the increased power to detect genetic associations between genetic variation and gene expression when we get down to more granular layers of information like single cell types compared to bulk tissue sequencing. So we performed co-localization analysis across the full mixed disease case and control data set using these 41 distinct CNS disease behavioral and structural brain traits. And in total, we identified over 500 co-localizations with a high posterior probability across 30 CNS phenotypes. And each of these co-localizations can be thought of as a gene cell type trait triplet. So in this upset R plot, the vertical lines here are situations where we find genes in common across different phenotypes. So we found 7 genes, for example, were shared between IQ in healthy populations and risk of schizophrenia. And we think that this might begin to explain the monotonic relationship we see between IQ and schizophrenia clinically. We also found 2 genes that were shared between Lewy body dementia and Alzheimer's disease, again suggesting perhaps convergent mechanisms for what are traditionally seen as divergent or distinct neuropathological neurodegenerative diseases. So these results can be interpreted a bit more clearly if I give you an example. So these are the 29 co-localizations we identified for Alzheimer's disease. Several of these co-localizations were present only in single cell types. So BIN1, for example, and PCARM, which are 2 very well-established risk genes for Alzheimer's disease, co-localized only within microglia, suggesting that that microglia may be the mediating cell type for those genetic effects on susceptibility to Alzheimer's. EGFR is another one here that was co-localized solely within astrocytes. And you can see here that the SNP maximally associated with the expression of EGFR in astrocytes is the same lead SNP for lead genome-wide significant SNP, conferring risk to Alzheimer's disease at that locus. So that's all well and good, but we wondered whether the use of disease samples might create a problem in a situation where disease itself alters the genetic regulation of gene expression. So the causal situation that, of course, we want to study is shown here, where we have genetic variants influencing the expression of genes in single cell types, and that relative expression mediates the relative risk of developing a disease. And we know that that SNP gene expression relationship is our EQTL. However, we might have a situation alternatively where we have genetic variation that confers risk for a disease, and then that disease itself causes secondary changes in gene expression. And then if the sample size is large enough, then we would still potentially observe an association between genetic variation and gene expression, and that would be measured as an EQTL. The problem here, of course, is that that EQTL has absolutely nothing to do with disease causality. So we wanted to understand what the relationship was of disease status to genetic regulation of gene expression. And to do this, we used this statistical interaction model where we took the base model for EQTL discovery and compared that with a model where we had an interaction term between disease status and genetic and SNP, genetic variation. And then we asked which of these 2 models best explain the data. And what we found was that depending on cell type, between about 15 and 30% of E genes had a significant interaction with disease status. And that can be seen more clearly here, where if, for example, we look at the relationship between this SNP and the expression of this gene in microglia, so looking across all of the mixed disease case and control samples, we see that we've got a significant EQTL association of 10 to the minus 6. But when we look at the individual cell types, we see that this association is almost entirely being driven by the Alzheimer's disease samples. So we thought it was sensible to only proceed with the control samples. And as I mentioned, these were no history of brain disease and neuropathologically normal brains. Because that control cohort data set was small, it was only 183 brains compared to nearly 400, we obviously discovered fewer E genes, so 7,000 in this data set on its own, compared to the over 15,000 from the full data set. And that translated to the discovery of fewer co-localizations, so in total 256 co-localizations across 26 traits in the control samples compared to 500 across 30 traits in the full mixed disease case and control data set. But what was really interesting for us was that 91 of these co-localizations were only present in the control-only data set, despite the substantially reduced power of this data set for EQTL detection. And that suggests that the use of disease samples can be obscuring some potential putative causal associations. So one example of this is shown here. So here we have a co-localization between the genetic regulation of this gene, PEX13, in excitatory neurons and genetic risk to multiple sclerosis. And we see this in control samples, but that co-localization is completely absent in the larger mixed disease case and control cohort. Conversely, we also saw co-localizations which were only present in the larger cohort compared to the control cohort. So for example, here we have a co-localization between genetic regulation of this gene in excitatory neurons and genetic risk to frontotemporal dementia. And when we look at the individual so if we take this lead EQTL SNP and this genome-wide significance SNP for frontotemporal dementia, we see that in this case, the EQTL association is being driven by Parkinson's disease samples. So we went on to perform two-sample MR using control-only sample data with a careful selection of the instrumental variables. So we had 183 control brains. We performed co-localization. We have a strict cutoff for defining co-localization. And then we select the instrumental variables. And to do this, there are a number of approaches you can take, but in the end, we decided that this felt the most appropriate. So we took the lead co-localized EQTL SNP from the co-localization. We removed the SNPs that were in linkage disequilibrium with that lead co-localized SNP so that we identified independent genetic variants to avoid inflating the MR statistic. And what we found was that, and then we reassessed the relationship between the instrumental variable and the gene exposure using the F statistic. And we found that following this, we retained typically one SNP as the instrumental variable for over 90% of the gene cell type outcome triplets. And then we went on to perform two-sample MR. And so in total, we identified significant MR evidence for an association between genetically proxid transcript levels in single cell types and a clinical outcome for 95 genes across 24 CNS traits. And these represent 142 gene cell type trait triplets. And so this can be perhaps better illustrated here by reference to Alzheimer's disease, again, where we have the 8 genes which had a significant association between the change in the level of their gene and risk of Alzheimer's disease. And I plotted them here according to their effect size, but also the directionality of the MR beta coefficient. So when the MR beta coefficient is positive, then that can be interpreted, because we know the directionality of the relationship for both the GWAS and the relationship between the genetic variant and gene expression, we can align this in a way that allows us to interpret the result from a therapeutic point of view. So if we have a positive MR beta, then that suggests that it's an increase in the level of the gene that confers an increased risk of disease. In contrast, if the MR beta is negative, then that is the opposite, so an increase in the expression of the gene would confer a decreased risk of disease or vice versa. So when the MR beta is positive, we know that we need to inhibit that as a candidate target, but if it's negative, then we know we need to activate that as a candidate target. So after doing this, because Arun will tell us that most drug targets are still proteins rather than RNAs, although I think that's beginning to change a bit, we wanted to see how many of these associations were validated at a protein level. And so for this, we used two publicly available data sets, the UK Biobank Pharmacogenetics project data set, which assayed nearly 3,000 proteins in 54,000 genotyped UK Biobank participants using the Olink Explore platform. And for brain proteins, we used this data set from Thomas Wingo, where they assayed 7,300 proteins in 330 post-mortem brains, and those were assayed using mass spectrometry. So from looking at the UK Biobank data first, of our 95 genes which had evidence connecting a change in the level of that gene with disease risk, 15 of those, representing 20 gene trait pairs, had a measurable protein in plasma in the UK Biobank data set. So of these 20 gene trait pairs, we selected instrumental variables based on plasma pQTLs using the same approaches that I've just described. And then we found that we could select instrumental variables for 16 of those 20. Then using two-sample MR, 11 of those 16 pairs, or nearly 70%, had a significant MR association between a change in the level of the gene's protein product in plasma and the same CNS outcome identified by single-cell MR. And I think that this really highlights the potential for this approach to inform plasma-based biomarkers that can be predictive of a drug response. When we looked at in brain, of the 95 MR genes, 44, representing 50 gene trait pairs, had a measurable protein level in the brain. And of these 50 gene trait pairs, we were able to select instrumental variables based on pQTL data for 21. And of these 21, 20, or 95%, had a significant association between the change in the level of that gene's protein product in the brain and the same CNS outcome identified by single-cell MR. So I think here it gives us a lot of confidence that the gene expression data, at least at single cell type level, is a good proxy for protein levels in the brain. So one example of this is shown here, where from our data, where we infer this protein GPNMB to be a candidate drug target for disease modification in Parkinson's disease. So looking first at our single cell data, we find that there's a strong co-localization between the genetic regulation of GPNMB in oligodendrocyte precursor cells and in astrocytes and genetic risk to Parkinson's disease. And then following selection of the instrumental variables and conducting two-sample MR, we find that there is a highly significant association between change in the level of GPNMB in these cell types and risk of Parkinson's. And the MR beta is positive, suggesting it's an increase in GPNMB that confers in these cell types that confers an increased risk of Parkinson's, meaning our therapeutic strategy would be to downregulate this gene. And then this was replicated using UK Biobank data and also using the Wingo brain proteomic data. We couldn't repeat the co-localization with that data set because of missing data. And so subsequently, it turns out functionally that GPNMB acts to regulate the uptake of fibrillar alpha-synuclein across the cell membrane, and that's thought to be an initiating step in the development of Parkinson's neuropathology. And this year we were awarded a grant, a drug accelerator grant from Parkinson's UK to work with the nucleic UK's Nucleic Acid Therapy Accelerator to develop an antisense oligonucleotide therapy targeting GPNMB as a candidate treatment strategy in Parkinson's disease. So this leads me on to my nearly final slide, which is to mention a large public-private initiative that's about to kick off called modestly the Landmark Consortium with myself and Verena as PIs. And this is a study that's really designed to develop the multi-omic data sets that we need to do this type of work at scale and really try to address problems related to horizontal pleiotropy through the generation of really dense and rich data sets with much greater coverage of gene expression regulation across many more cell types. And so here we'll be generating single-cell transcriptomics, proteomics, metabolomics, and lipidomics across thousands of genotype brain samples. And each of these brain samples, importantly, will be linked to state-of-the-art digitized neuropathology, including quantitative measures of oligomeric tau and alpha-synuclein, which will allow us to begin to look at mechanisms of disease progression as well as disease susceptibility. So I hope I've shown you that this sort of data can provide the framework for target and predictive biomarker discovery. Importantly, we get information on the directionality of the relationship, and that's vital for informing therapeutic strategy. We get information on potentially mediating cell types, again important for drug development, high levels of replication from proteins, which is reassuring. And then just to acknowledge the funding, mainly from the MRC, some funding from Roche, and the amazing team that we have with Verena, Leo Butolo, who works in Cambridge, and a talented PhD student, Alex, at Imperial, who's hopefully staying on with us this year. Thanks very much.
Thanks very much for a great talk, Michael. As we're approaching the tea break, I think we have time for just one question at this stage. Yes, at the top.
Two questions, actually. First one, do you use phi mapping to select variants? Second, how do you interpret the S single-cell MR from healthy controls? Do those genes relate to the disease or not?
So I didn't catch the first question, but the second question about MR, strictly speaking, one shouldn't be conducting MR in disease cases. But clearly we're seeing co-localizations in disease cases which are not present in controls. And I think that that's super interesting. So there's a situation with ALS and frontotemporal dementia where the genetic variants in UNC13A affect the inclusion of a toxic transcript into TDP43. That inclusion of the toxic exon into TDP43 is only seen in disease cases. So I think that there will be situations where we're looking at disease causation, perhaps over decades in a normal brain, but we're also be situations where genetic risks for certain brain diseases will only manifest in the context of pre-existing pathology. And that kind of makes sense given that we know that the prodromal periods for development of diseases like Alzheimer's and Parkinson's is over decades. So I think if that risk period is long enough, then from a GWAS point of view, they will still appear as genetic susceptibility loci rather than disease-modifying loci. So from my point of view, I think it's really important to look at both disease samples and control samples because they tell you something slightly different.
Okay, I think we will end the session now as we're 10 minutes over time. No, it was very interesting to hear this. So I'd like to thank all the speakers in this session and to remind everyone that the next session, the final session of the afternoon of 2 more talks, starts again at 3:30. It can be 3:40. 3:40? Okay, let's come back at 3:40 then. Sir Munir has kindly given us some extra time to enjoy our tea. Thank you.
Okay, Good afternoon, everybody. I hope you enjoyed your afternoon tea. I think Munir's still enjoying his afternoon tea. And just to say a big congratulations to Munir and the network and BPS for organising such a fantastic event today. Really enjoyed it myself, really sort of learnt some new things as well today. And I've been particularly interested in sort of all the drug discovery and the medicines area. So my name is Karen Spink. I am Head of Medicines at Innovate UK. We are the UK's National Innovation Agency. And so I'm interested in all things medicines, therapeutics, and medicines manufacturing as well. So my great pleasure to introduce this session on new technology platforms. And I think what we've heard about today are sort of around these new tools that we have now that are available, and also an update on all the cohorts, the population and the patient cohorts. And I think there's just a huge amount of opportunity there to really focus on improving our access to the data and improving our tools for actually interrogating that data and getting the best from the data. And I think we're going to hear a bit about that in the next talk. So a great pleasure to introduce Greet De Baets. And Greet is the Director of Data Science from AstraZeneca in Cambridge. And Greet will be talking about AI target identification and patient stratification. So, okay.
Yeah, thank you for the introduction, Karen, and thank you for the invitation to present here. So as Karen said, today I'll talk a little bit more on how we within AstraZeneca use advanced technologies to really make precision medicine from the start possible. I will touch on different topics, so I'll focus on single cell analysis, but also knowledge graphs to drive target identification, and I'll also touch on some traditional machine learning approaches to drive patient endotyping. However, before I go there, I'll first discuss a little bit more on why precision medicine is really important in the respiratory and immunology field. So, because that is really our remit. We really want to make precision medicine a reality because we're not there yet in respiratory and immunology. So, for decades, scientists in a way have been studying the causes of disease. And as discussed today, we know that precision medicine has really revolutionized the treatment of some cancers and rare diseases. However, for chronic diseases like precision medicine, which we define as matching the right medicine to the right patient at the right time, is not that common, even though we know that these diseases are biologically very complex and driven by multiple mechanisms. And this is, for example, clearly illustrated in this example where we looked at the FDA approvals over the last 20 years in asthma and COPD. For example, in asthma, from all the FDA approvals that have happened over the last 20 years, only 4 of these target, in a way, novel mechanisms in asthma, and we only target 1 novel mechanism in COPD. And this is, in a way, kind of surprising, given, again, how we know how complex these indications are. For rheumatoid arthritis, the problem is slightly different. So we have multiple approved drugs targeting different mechanisms, but there's an opportunity for disruption given the response that is observed. So the current treatment paradigm is that patients will start on methotrexate, and then dependent on the response, they will move on to biologics. And they will cycle through these different biologics if no response is observed. However, this choice of treatment is in a way not driven by molecular characterization of the patient. So it's very ad hoc at the moment. And we really think that if we want to improve the patient outcomes, we need to move beyond a one-size-fits-all approach and move towards the possibility of targeting treatment to the specific disease states. So to achieve this, we within the department focus on 3 main buckets. So we want to identify the right targets, the right patients, and the right biomarkers. And in this talk, I will talk a little bit more on how we apply advanced technologies to drive the identification of new targets, and to also get a better understanding of the patient heterogeneity. I think the previous speakers have already clearly illustrated that there's a real challenge in identifying good targets. So the methods that I'm going to discuss today are a little bit more non-traditional in a way. So the first one that I'll touch on today is identifying targets through deep molecular characterization of the disease tissues. And the focus is really here on disease tissue, given that we know how important it is to look at the local disease biology and not necessarily at systemic effects. And I think the previous speaker, again, clearly illustrated the importance of doing this. So the data type that we're going to use for this is single cell data. And the use case that we'll focus on is with an inflammatory bowel disease. At the moment, within the IBD space, we're in a way not as lucky as the previous speaker. So we don't really have very large cohorts as we speak, for which we have single cell data analysis. There are many different public studies out there which generated single cell data from the tissue from IBD patients. And that was either with the remit of predicting the response to a treatment or for the novel target discovery. However, all of these individual single cell studies are quite often underpowered. And that's why we are collaborating currently with ImmuneAI to generate this integrated, harmonized single cell atlas. And this integrated cell atlas will enable comparative analysis across the cohorts by providing a harmonized cell type annotation, a higher resolution cell identification. We will apply batch correction and, of course, create the metadata. And it will be this single cell IBD tissue atlas that we will use as the data foundation for novel target discovery. So to get a little bit more into detail. So we harmonized 20 different single cell studies, and these covered both inflamed and non-inflamed samples. We also had data from patients that responded to anti-TNF treatment or not. And this whole atlas, in a way, comprised, in a way, around 1 million cells. Again, quite small compared to the previous speaker, but quite big for us at this point for IBD, across 200 patients. And given that we have harmonized this clinical metadata, we can now really start to extract signals across these different patients. It's also important to show the granularity that we have achieved across the atlas. So the atlas contains 76 cell subtypes. And here you notice, for example, how this differentiates from the original author's annotation. So for example, for this particular study, the power of the data set only allows the authors to annotate the cell types onto mononuclear phagocytes, but then by harmonizing all of them together, we could really go into more granularity and annotate these at a more fine-grained level. So after constructing this IBD tissue atlas, which is represented on the left by a UMAP plot, we applied some advanced methodology. And we used ImmuneAI's proprietary representation learning method to represent individual samples in this multidimensional latent space. So each dot here does represent a cell anymore, like on the left plot, but each dot in a way represents a sample now, where the color indicates the biological condition. For example, an inflamed sample, a non-inflamed sample, a responder, a non-responders. And it's from this sample level latent space, we can now identify key cell type specific transcriptional features, or simply stated as gene sets, that allow differentiation between the subgroups. So for example, which features of interest allow us to differentiate between responders or non-responders, or allow us to differentiate between inflamed versus non-inflamed. And once these features of interest are identified, we can identify or extract the genes from these features. And we'll do that by having a closer look at the associated weight for each of these genes to the feature. This initial set of genes will then need to be further triaged based on atlas-derived data, but we will also include other metrics. So we'll include genetics data, we will include pathway evidence, and we'll also look at some commercial data or druggability data. And this will then result in our target shortlist. Of course, with any AI method, there's of course first some skepticism. We need to grow confidence in the methodology. And that's why we wanted to have a look at how much of the known IBD targets are covered by this initial list of 400 predicted IBD targets. And it was really reassuring to observe that within the top 400 genes, we captured targets representing 5 of the major therapeutic pathways in IBD. So this really increased our confidence that these targets will result in a way in clinically viable targets. Another way to grow that confidence is by looking at the biological pathways that are enriched in the features from which these genes are derived. And again, we noticed that a lot of the pathways that are picked up have established importance in IBD. And it's also very reassuring to see that we pick up on biological diverse pathways. So both of these investigations, in a way, increase really our confidence in the usability of this approach. However, it doesn't stop here. As discussed again before, this is just the start. Now the really hard work comes. Because now we have predicted targets, now we need to go and functionally validate them before we even can think about bringing them into our drug discovery pipeline. And the way that we will do this is we will do this through functional genomic validation. So we'll do this through CRISPR screens. And the first step that we'll need to do is identify the relevant in vitro model system in which we want to do the CRISPR screen. And again, we will use the IBD tissue atlas here to measure the transcriptional similarity between the cell subsets and the experimental systems that have been developed. So once we prioritized our experimental system, we will then knock out these targets into this system and measure the functional changes via proteomics and transcriptomics changes. And as such, we can again evaluate which targets might have the most clinically functional effect. And finally, we will then use the IBD tissue atlas again to interpret the disease relevance of the candidate targets by back-translating the functional genomics results. And again, if any of these hits will be promising, we will take them forward into our drug discovery pipeline. So this is really only the first step. Another method that we use for target identification is using knowledge graph. And the aim here is really to identify novel, less obvious targets. So let's first start with what a knowledge graph is. So a knowledge graph is in a way a representation of relationships named edges between objects called nodes. And we can generate a knowledge graph across different domains. So it doesn't really have to be biology. So the examples that I've shown here is shown the betrayals in Game of Thrones, where in a way the node size indicates the number of betrayals, and the edge weights indicate the impact of the betrayal. So as you see, a knowledge graph can really be applied anywhere. And within AstraZeneca, we use it for multiple use cases. And I've mainly been using it for target identification and lifecycle management purposes. And of course, to drive these use cases, we need specific data. And the data that we need to include is commercial data, feasibility data, safety data, the data that is coming from our clinical studies, but also we really need to make sure that we capture the molecular biology in a good way. And this subgraph is where I will spend a little bit more time on today. So within this biological insight knowledge graph, we really represent the key biological concepts and how they are connected. So the key relevant biological entities are gene, disease, compound. And for example, a gene will be connected to a disease if there's known evidence linking it with the disease. So this can be either literature evidence, but it can also be genetic evidence or up-regulation or down-regulation of that particular gene in an indication. A gene will also be linked to a pathway or biological process if it participates with it. Genes will be linked with each other if they regulate or interact with each other. So this knowledge graph should really represent how we think about molecular biology at this point in time. However, as again discussed already earlier today, I think that's one of the benefits of speaking later in the day. A lot of topics have already been touched on. The quality of the data is really key for any method that we apply to. And the same applies to knowledge graph. The predictions that we will get will only be as good as the data that is included within the knowledge graph. So we really need to avoid that people think of a knowledge graph in a way as a data dump. So that's why we spend some time into enriching our knowledge graph with relevant data sets and also curating the data sets that have been included. So we've integrated or enriched the knowledge graph with some internal data. So we have a lot of internal data where we have tested our different compounds in, again, different model systems, and as such assessed whether a gene is up or down regulated about compound treatment compared to control. We also work together with our clinicians to really curate the disease ontology, because I think that's, again, something that a lot of improvement can happen in. And we also integrated some single-cell data, given that we know the importance of looking at the expression at the relevant cell type, at the relevant tissue. So once these knowledge graphs are set up, we can apply advanced methodologies on them. And one method that we've been playing with is autoencoders. And the aim of this method is a way to learn a low-dimensional representation or embedding of our input space. And this is a method that is commonly used in imaging, so that's why I added an illustration of Leo the cat. So an autoencoder, in a way, consists of two main steps. An encoder that will generate a lower dimensional representation of the input image, and the decoder that will try to reconstruct the original image using this representation. And this decoder step is really necessary to build confidence in our representation, as if we're unable to reconstruct the original image, we also can't really trust the representation. And a similar methodology can in a way be applied onto knowledge graph, where the encoder will generate a representation for every node, and the quality will be assessed by comparing how the reconstructed graph compares to our input graph. And that's exactly what we've done in a way in our collaboration with Benevolent AI. So using their proprietary methods, we've generated our knowledge graph predictions, after which we again had to triage the targets and prioritize them based on the biological evidence and the feasibility. And then these prioritized targets were assessed into CRISPR screen, followed up with a smaller scale experiments for the CRISPR hits. We've applied this knowledge graph target identification onto multiple indications, but I'll focus on idiopathic pulmonary fibrosis. And here we show an example of target 1 in a way. And it's clearly from the picture on the left, you see that there's limited direct evidence linking this target to the indication. So this target would never have been picked up by the more traditional measures like gene expression or literature mining. So when we look at the indirect evidence, however, it's clear that this approach is really good in surfacing the targets that are enriched in indirect evidence. So for example, this particular target is linked to IPF, through the biological processes that are linked with IPF, and it's also linked with related diseases as IPF. So again, this was the first step, like the computational method. Now the experimental validation comes, and that's where the real power lies. So given that the indication that we focused on was idiopathic pulmonary fibrosis, we validated our hits in a lung fibroblast CRISPR screen, where we look at TGF beta-induced fibrosis. So if we have a look at the plot on the left, the different dots in a way represent the different gene knockouts. And on the y-axis, you see the percentage of TGF beta-induced fibrosis. And you see that the blue dots, they are the untreated conditions, and the red one are the TGF-treated conditions. And you see that for most of the gene knockouts, these still induce like a high percentage of fibrosis. So these are not the targets that we're after. However, there are some hits that clearly inhibit the TGF-induced fibrosis. And these are the targets that we really need to have a closer look at and investigate into more detail. So on the right, you see a little bit more close-up on how the plot on the left was generated. So in the top row, you just see a control condition where you have the untreated condition, and you see that alpha-SMA is really expressed upon TGF treatment. On the row below, we have our control gene. So this is a TGF-beta receptor knockout. And we indeed would expect that in this knockout, we don't see the fibroblast activation. And this is indeed true. We don't see like this overexpression of alpha-SMA in the second row. However, in the third row, which is the target 1 knockout, we see a similar effect as our control, which again tells us that this might be a hit worth pursuing. So this gene is now further looked into through more specific assays. And again, if these are successful, they will be taken forward into our drug discovery pipeline. So once these targets progress through our pipeline, we really need to start thinking about which patients will benefit from this particular treatment. And that's why it's really important to get an understanding about the different patient endotypes that drive heterogeneity. And quite often you will see that patient endotyping efforts starts using molecular data, so either protein expression data or gene expression data. In this section, I will showcase another approach where we use comorbidity data as a starting point to cluster patients. And the underlying hypothesis into why we look at comorbidity data is the fact that genetic drivers of disease may be active in multiple organs, and as such manifest into different conditions. So if we cluster patients by their comorbidities, it may reveal groups that have a higher incidence of rare variants that drive a particular disease. So we do this type of analysis by looking at the UK Biobank, and more specifically at the COPD patients in there. So we have around 18,000 COPD patients in there. And a first interesting finding is that approximately 1/3 of the COPD patients in UK Biobank have more than 2 reported comorbidities. And what is really interesting for us as AstraZeneca is that these comorbidities align with other indications that fall within our strategic indication that we look into. So besides respiratory and immunology, we also focus on cardiovascular and renal indications. So this is really a good guideline that might help us in repurposing existing therapies that we already have. But to cluster now the patients using the comorbidity data, we'll first generate a comorbidity space by indicating for every patient whether they have a particular disease or not. So on the left, the row in the way represent the different patients, and the columns represent the different diseases, where a Boolean will indicate whether a patient has the indication or not. And this was then converted in a 2-dimensional map of comorbidity by self-organizing maps. And each hexagon now represents approximately 30 subjects, and adjacent hexagons represent similar subjects. We can then, for example, use color in the space to represent the prevalence of a selected comorbidity. So in this case, we look at hypertension, where blue indicates a low prevalence and yellow points to a higher prevalence. And again, this confirms that similar subjects are really grouped together. In this slide, we show some more comorbidities, and you clearly see that the different clusters, which are indicated by the white lines, are enriched in different comorbidities. To identify now the comorbidity clusters that are enriched with COPD patients, we also have to place non-COPD subjects into this map using the same comorbidity description. And by comparing the odds ratio of cluster membership between COPD and matched non-COPD subjects, we can identify which clusters are enriched for COPD patients. And it was really interesting to see that we find no enrichments in the overall healthier groups, but the multiple comorbidity clusters are really enriched up to 5-fold into COPD patients. And to us, this really shows that this unbiased comorbidity clustering can be used to identify trends in large cohorts that contain comorbidity data, and as such highlights new potential patient subtypes. And identification of these different subgroups can then be considered in a way as a starting point for target identification with precision medicine from the start in the back of our minds. So just to quickly summarize, so by clustering patients by their comorbidities, we really hope to identify the genetic drivers within these clusters. And as a next step, we will, of course, leverage knowledge graph and functional screening to triage the hits for their relevance. I hope that with these 3 examples, I have showcased how we are using advanced methodologies to drive target identification and make precision medicine a reality in respiratory and immunology. And to conclude, I really would like to thank everybody who contributed, as the work that was presented was led by multiple different people and different external collaboration. Thank you for listening.
Thank you, Greet. I thought that was fascinating. I think it's always really helpful to have an exemplar that we can focus on and see things in reality as well there. So are there any questions for Greet? Munir.
Thanks very much. That's a great talk. Just wondering about your knowledge graphs, and you had safety as one of the aspects, but using CIDR for the safety database, and MEDRO, which is a coding database. But CIDR is pretty awful, actually, so your ability to be able to identify any adverse effects through your knowledge graph is going to be very limited. So do we need much better safety databases for you to be able to do something like this?
Definitely. But I don't think it only applies onto safety data. I think it applies on any of the sub-knowledge graphs that we have. Commercial data sets has, in a way, similar challenges. I think one of the main challenges, for example, that we face is how we annotate in indications. There's no harmonization across which disease ontology to use, which makes it, again, really challenging to bring all the different data sets together. So yeah, any help here is appreciated in making these better.
Thank you. Can I just ask a quick question on the clustering of the multimorbidities? So you talked about repurposing, so obviously that's a really interesting angle. But do you think there's an opportunity there for prevention as well, where we can identify those patients who might benefit from earlier interventions?
We haven't specifically looked at that question. Again, given all the other challenges that have been discussed here about like the timing of when indications happen, but it's definitely something that we can look into.
Brilliant. I think there was one question over there.
Yeah, I also want to go back to the knowledge graph and 2 questions. One is, I mean, the example is really nice you show, but have you benchmarked this on a broader set of like true positives or whatever? And if so, have you ever used any explainable AI techniques to try and understand why the graph is prioritizing this over others?
So to first answer your first question, so Benevolent AI has published their method than they're benchmarking a few years ago in the public domain. So that publication is out there. It's always a challenge with benchmarking target identification efforts because, for example, if you rely on literature, once a target has been identified, you see a rise in evidence for that. So it's like finding this right threshold at what data you look into for your benchmarking. But it's something that they've definitely looked into. Your second question, I forgot now.
It's about, I mean, these deep learning tools are cryptic and black box whatsoever, but there are now more and more techniques to demystify them, trying to look at attention scores or whatsoever to try and understand why do they do these decisions.
So again, benevolent AI has two methods in a way, and that was one of the main questions that we had, because we didn't just want to take the target that they suggested at face value. We needed to get an understanding on why they were selected. So that's why they have built a whole dashboard on top of their knowledge graph to really surface why particular projects were prioritized and what is the underlying evidence that is present in the graph to really support that evidence. We definitely need explainable AI. We need to try to move away from the black boxes, I think, to really trust these predictions more.
Okay, thank you very much, Greet.
Thank you.
So our next speaker is Professor Julian Knight. So Julian is Professor of Genomic Medicine at the University of Oxford. And Julian will be talking about infectious disease, individual response to infections such as sepsis and development of autoimmunity. So over to you, Julian.
Thanks very much. It's a pleasure to be here. I'm very grateful to Munir and the organizers for the opportunity to tell you a little bit about some of our experiences of trying to apply a stratified medicine approach to understanding the clinical challenge of patients with severe infection. For those of you that aren't particularly interested in infection, I think that a lot of the approaches that we apply are broadly applicable across other diseases, and I think that's a theme that will have been running throughout the day. So, the sort of work that we've been doing obviously had publicity recently when we all became aware of the importance of a pandemic and just how we start to recognize why some patients become very sick and how we can manage those patients. But the clinical syndrome that I'm going to spend a lot of time talking about today is essentially these patients who have a very severe response to infection, and that's the clinical syndrome of sepsis, and that's been with us for many years and remains a major challenge. And a lot of this is about the heterogeneity of trying to have a basket in terms of a clinical syndrome, or indeed a particular microorganism. And how can we start to think a bit more about some of the underlying disease processes and how we might target those? And the reality, of course, when we're thinking about infectious disease is that all of us are exposed to pathogens, and almost all of us respond in an appropriate way if you think about your immune, inflammatory, metabolic response that restores homeostasis and effectively gets rid of the pathogen. The challenge that we're facing is how we deal with the small minority of patients for whom that is not the case, and in whom that response might be insufficient or maladaptive. So we can think about a failure of response in terms of people that might have a primary inheritable cause, for example, a primary immunodeficiency, or they may have an acquired immunodeficiency. And then there's a broader group which we understand much less well, where this response is somehow inappropriate or maladaptive. And there are various words or terms that people use. You can think about having a hyper-inflammatory or a hypo-inflammatory state. You might talk about having a cytokine storm, evidence of immune dysregulation in a particular immune cell population. There might be more general features of immune suppression. We talk about T cells being exhausted, they're refractory to repeated stimulation. And some of this begins to overlap with what we see and understand increasingly in the setting of aging, where after the age of about 70, our immune systems become really quite inefficient, and these ideas of immune aging and inflammaging start to overlap with the challenges that we see in younger patients as well as older patients who are not responding appropriately. And this has profound consequences for your ability to survive the acute infection. But if you're discharged from intensive care after this very severe episode and you recover, your mortality rate and your morbidity unfortunately remains very high. So we need to do something for the survivors of severe infection, including those with long COVID, where we think there's underlying immune dysregulation that might be driving this. So for a long time, if you tried to convince a pharma company to do a clinical trial in a syndrome like sepsis, they wouldn't do it. And the reality is that we've had no immunomodulators that have been successfully approved and have lasted more than a couple of years in clinical practice. This is a real challenge, and we need to identify the group of patients that are going to actually benefit from these interventions with much greater precision. And some of the tools that you've been talking about today, particularly in terms of molecular stratification, offer us opportunities to do that. And I'm going to particularly tell you about application of transcriptomics and proteomics, but there's all sorts of layers to this type of information, as you're all too well aware as an audience, which range from germline genetics through epigenetic processes, ways that we can measure that at the RNA, or changes in histone modifications or accessibility, ways that we can map those differences between individuals. And then perhaps the biggest challenge that we all face in this field is how you combine that information and make the most of it. And I'll touch on that, particularly in the last part of the talk. So I'm going to tell you about how we've been applying particularly whole blood transcriptomics to try and identify a particular subgroup of patients with sepsis. Some work that we published in the last couple of weeks where we've been applying a proteomic approach, again, towards patient stratification, and then how we've been leveraging multi-omics data in the setting of COVID-19, more about this challenge of how you maximize the value of multi-omics most effectively. So the work published in The Lancet showed that in 2017 there were 49 million cases of sepsis worldwide, 11 million deaths. This is a global health priority. It applies to the UK. It's the most common reason that you might be admitted to a medical care, intensive care unit. And it has a poor mortality acutely of about 20 to 28%. And this is a term that's been used for a very long time, but actually it was William Osler who perhaps had some of the insight that we now are accepting more broadly, where he said that except on few occasions, the patient appears to die from the body's response to infection rather than from it, it being the infection. And so this is the idea that's now captured in the consensus definition of sepsis from 2016, which is of life-threatening organ dysfunction caused by a dysregulated host response to infection. And that's great. But if at the bedside, if you were to try and make that diagnosis, we're essentially relying on recognition of the patient having infection or very high clinical suspicion of infection and evidence of organ failure, which might be a single organ or multiple organs. We don't have the biomarkers to tell us about this underlying immune dysregulation. And so these consistent failures in clinical trials had really put off pharma companies, but I'm pleased to say that there's renewed interest now. And that is because of the success of immunomodulatory therapies in the setting of a more homogeneous patient group. In other words, patients with severe COVID-19, where we know that interventions like dexamethasone, modulating interleukin-6, JAK inhibitors do show benefit. I'm not convinced that we've totally solved the question of how you define the group of patients that are going to benefit most significantly, but nevertheless, clinical trials such as Recovery have demonstrated this and have really been transformational. So there have been a variety of reviews published. This is a very nice one from Tom van der Pol and Manu Shankar, which capture some of the different mechanisms that people have proposed in terms of how sepsis might be driven, and we'll touch on these in a bit more detail. But essentially, we don't know in which patients a particular mechanism might be predominant, or indeed at what stage of the illness. And so the work that we published some time ago now was based on a study that recruited patients with community-acquired pneumonia from intensive care units across the UK, where we did whole blood transcriptomics based on microarrays at this time. And we took an unsupervised approach to the data. We said, what clustering might there be in this data? And we found that there was evidence of subgroups, as you might expect. And there were at least 2 major groups, one of which showed a significant association with mortality, which we became more interested in. So what we're talking about here are sepsis response signatures, and it was possible to define a relatively small number of genes that would predict your membership of that particular subgroup of patients. And this was initially, as I say, in the terms of pneumonia. And in a subsequent paper, we showed that if you looked in patients with fecal peritonitis, a very different infectious etiology, you saw a very similar phenomenon whereby the patients separated into at least 2 groups, one of which was this poor outcome group. And so for this audience, you might be interested in this kind of principal component analysis plot. So explaining the majority of the variance is principal component 1, and this is the second component explaining about 10% of the data in terms of variance. And if you look at these sepsis response signatures in red and blue, you see that they're really being pulled apart in this first principal component rather than whether the circle is filled or unfilled in terms of the underlying infectious driver. And what we've shown across a variety of infections is that this paradigm seems to hold up in terms of a particular subgroup of patients we can define with a relatively small number of genes. We then did some work with Tony Gordon and David Ancliffe at Imperial College, and they were able to work with us to do a retrospective analysis of a clinical trial which had involved use of steroids for patients with septic shock. And by analyzing, as I say, this is a retrospective analysis, the patients in terms of their outcome and assigning the particular subgroup that they were members of based on this predefined set of 9 genes, it was possible to show in the patients that were receiving placebo, there was the same relationship with the poor outcome group of patients with SRS1 versus those in the other subgroup. And these patients showed no difference in terms of hydrocortisone, but there was actually evidence of potential harm in the other group. So this is very preliminary data, but I think it shows the opportunity to be applying this sort of subgroup analysis in the setting of treatment decisions. More recently, we've been taking this idea and thinking about how, rather than putting patients into a particular group, we can think about the likelihood of being members of that group, and also try to anchor this data using information from healthy donors. And so we've used large data sets both based on microarrays and RNA sequencing from patients with different infectious etiologies of sepsis and healthy controls. And in a similar way to that principal components analysis plot, start to think about this continuum where we move to this particular poor outcome group of patients on the left here, and we start to be able to define a quantitative score, the SRSQ score, which we can then apply and think a bit more about how that might start to be something that you could use clinically. And so we've established a machine learning framework where you can put a single patient's data into this trained data set and establish a likelihood of being in this particular patient subgroup based on your SRSQ score. And this is just showing you a little bit of data from COVID-19. So here's the SRSQ score, and we can look across different WHO-defined subgroups of COVID-19 from mild, severe, or critical, and see a similar relationship in terms of severity and mortality. And we're working with Cepheid, who have a multiplex RT-PCR-based assay in a cartridge that is used primarily for pathogens currently to try to see if it's possible within 45 minutes to be deriving an SRSQ score based on whole blood. Patients change in terms of their membership of this particular subgroup, and you can see that those in whom the score is changing over time have a very different relationship in terms of their likely long outcome from the acute illness. And we think that this is telling us about some of the host mechanisms that are happening in terms of underlying immune dysregulation and the likelihood of underlying and subsequent organ dysfunction and response to therapy. So this is, if you like, a hypothesis of what we think might be happening, and here's a little bit of evidence to support this. So you've already heard about single-cell work, and so Andrew Kwok, who is a very dedicated DPhil student, took this sort of single work to an extreme. So he was running a fresh sample individually to try and generate a single-cell profile, and the reason for running a fresh whole blood sample was we were particularly interested in neutrophils. And you need to do that because it's very hard to, for example, cryopreserve neutrophils. And so we did a single-cell multi-omics experiment, and what this showed was that there was evidence of particular dysfunction in more immature neutrophils that was very much enriched in this population of patients who had this poor outcome endotype with a relatively immune-suppressed phenotype. And what we think is happening in these patients is that they've got underlying dysfunction in the bone marrow. So as a way of trying to look at what might be happening in the stem cells in the bone marrow, we looked at circulating stem cells, and we were able to look at clusters of those stem cells. They're very rare, but it's possible to enrich them, and we found that in these poor outcome patients with a particular subphenotype SRS1, there was enrichment of a particular cluster of these stem cells, and evidence that I don't really have time to tell you about today, but particular transcription factors involved in emergency granular poiesis, such as CBP-beta, and other features in the data sets, and through some more immunological work that we did, such as to suggest that there's a dysfunction in terms of emergency granular poiesis that's driving this pathology and the phenotype that we're seeing. So it's come from an observation in terms of perhaps a somewhat crude setting of whole blood transcriptomics, but actually has allowed us to start to tease out a particular mechanism. And what we think is that actually having done that, there's an opportunity to revisit clinical trials that have been done in sepsis previously, where people have given, for example, interleukin-6 to all sepsis patients or tried to modulate it in some way. And rather than do that, if we can do targeted therapy to individuals in whom we have a clear biological rationale for why we might be doing that sort of immunomodulation, perhaps we might have more success. All right, so I'll just now tell you a little bit about some work with plasma proteomics. And as you've already heard about today, this takes us a little bit closer in terms of that paradigm from DNA to RNA to protein. And we think in the setting of sepsis may be particularly informative for underlying organ dysfunction if we were to look in the plasma, which will reflect not only the immune cells that are in the blood, but also products from major organs such as the liver. There's been a big challenge, as you're probably aware from earlier talks, in terms of how you do effective quantification at scale, and because of the large dynamic range, and work that we did with Roman Fisher in Oxford that was led by Yushin Mi, who a PhD student in the team, involved a very large number of patients that we were able to bring together, both with sepsis and a number of control populations, both healthy controls, but also patients with sterile inflammation, and patients who were before and after surgery who had no evidence of infection. And the approach that we took here was based on mass spectrometry. And we were able to work with Roman to increase the throughput on that. At the end of the day, we had a relatively small number of proteins that passed all of the QC, and this is a technology that's continuing to evolve, and you've heard already, I think, about technologies like O-Link, where it's possible to get a larger number of proteins. But nevertheless, it's an interesting cohort, I think, in terms of its scale. And I'm not going to be able to talk you through all the types of analysis that we've done, and you've heard about many of them today, but it's possible to also think about time series, as we had samples at different stages in the illness. But the focus I'm going to really talk about today is the opportunity to do this sort of unsupervised clustering. And so this is just showing you a little bit of the data in terms of a principal components analysis, and you start to see some sort of trajectory as we move across to the sicker patients with sepsis and some of these other populations that we were able to study, and look at the particular proteins that were contributing to the different principal component loadings. One way of bringing together this sort of information is to look at co-expression networks, so proteins whose patterns of expression are similar, and you can then think about particular contrasts, for example, between the sepsis patients and, in this case, the healthy volunteers in our discovery cohort, and look at what might be enriched in terms of particular biological pathways or processes in those groupings of proteins. And so thinking about this question of stratification, if we took the large number of sepsis proteins, so we had, patients, we had over 1,000 patients, we could look at how they started to cluster. There was evidence of probably 3 main clusters. It's possible, no doubt, to go into more subclusters, but the k-means analysis suggested that was a reasonable number to be using. And again, like we saw with the transcriptomics, you start to see differences in terms of these clusters in terms of survival over time. So you can think about the clusters then in terms of more refined measures of underlying organ dysfunction and the particular state of the patient, and we can get measures of this for different organ systems based on your SOFA score or sequential organ failure assessment score, and see the particular relationships. And essentially we found that one of these clusters, cluster 1, was associated with poorer outcome and was associated with particular proteins showing differential abundance in particular processes. And this is summarized here in terms of a whole variety of immune processes that appeared to be different in this particular cluster. So in terms of how that might relate to the earlier transcriptomic data, for the majority of these patients, we also had information on the transcriptome, and we could bring the two together. And it turns out that this is not overlapping. There is some overlap, but we're getting some distinct information from the plasma proteome when we combine it with the whole blood transcriptome. And the patients that are doing particularly badly are those that are in this proteome cluster 1 and also in the sepsis response signature from the transcriptomic group 1, shown in orange here. And we need to understand a lot more about this to really tease out the biology. So in the final part of the talk, I just wanted to touch on multi-omics. And this is data arising from a large consortium which we ran through the pandemic, where a lot of groups came together and worked on the same patient groups and samples, applying their particular expertise. So it was a remarkable opportunity, really, to bring together people who really understood information about applying single-cell approaches, looking at both the transcriptome and the proteome, but also epigenetics, understanding differences in the repertoire, applying sophisticated immunology in terms of mass cytometry. And we recruited patients in the community with COVID-19, patients admitted to hospital with different severities of disease, but then we also had comparator groups in terms of healthy individuals, people with sepsis recruited before the pandemic, and people with severe influenza. And we learned a lot through this, and I'm not going to talk you through the individual data sets, but more focusing on this part of the process whereby we start to bring the information together. And just to show you a little bit about how you can start to combine different types of protein data measured in very different ways. So this is protein measurements based on mass spectrometry, and then using Luminex assays, which pick up these smaller proteins that are not captured very well by this method, particularly cytokines and chemokines. And the approach here is to think about what network might exist within an individual data modality or assay type, and then with a second assay type or more, and then how do those networks relate to each other? In what respect are they similar or different? And Alberto was able to derive single similarity networks based on fusing this type of information and show that this captured a lot more information. And this is showing you a principal components plot, showing you how he subsequently identified at least 2 major clusters within this and the particular proteins that were different. So why is this interesting? Well, if you then think about this in terms of the way that clinically we were dividing patients, whereby based on WHO criteria, you might define mild, severe, or critical disease based on the strength of color of the shading of these dots. Actually, what this proteomic-based approach was doing was actually we can start to divide the severe patients who were actually an intermediate group into one or other of the groups. And that might help us in terms of defining when we're using particular interventions, when we're unsure of this cutoff between severe and critical illness. And we know that it's possible to do harm, for example, with dexamethasone as you start to treat patients with milder disease. There was a clear relationship with mortality with those patients on the right-hand side here. And we were able to take a small number of these predictive biomarkers and look with a completely independent proteomics data set generated at the Massachusetts General Hospital and generate data that convincingly showed a very similar relationship with mortality and outcome. So just finally, a more sophisticated way of combining data was based on work we did with colleagues in the Mathematics Institute in Oxford, whereby we took all of our data sets, which were high dimensional, and you can think about these on a per sample basis because they all came from the same patients. And a bit like doing a principal component analysis, you can do something called a tensor decomposition, which generates a matrix and defines components that explain that matrix. And each of these comprises vectors of scores from the different data types. And this allows you to think about how a particular component might combine information about particular cell types, about particular proteins, about particular genes, if you can then relate that component to a clinical feature. And so, for example, this is showing you a heat map where the strength of correlation is shown in terms of the colors with some different clinical features or demographic features. And this is actually showing you gender and showing how this particular component is being driven by genes, the level of gene expression on our sex chromosomes. But obviously, it's more interesting to then dig into some of these clinical features. And the component that we found that was most significantly associated with COVID-19 severity was this particular component, 171. This is showing you the score for that component in patients with mild, severe, or critical illness and those with healthy disease. And clearly, there is also overlap with what's happening in these other patients with flu and sepsis. So it's probably more about this idea of what's happening more generically in severe disease. And what this component is doing, based on that purely mathematical approach of tensor decomposition, is pulling out information that says these plasmablasts and a particular type of monocytes appear to be very important. These particular plasma proteins are highly predictive or involved, and then particular genes involved in antigen presentation and T-cell signaling. But it's interesting that these are all coming together with a particular component, and this is an approach that you could apply with any type of multimodal data set. So I think that these types of tool are very helpful in terms of understanding the complexity of these phenotypes, understanding shared and specific features, and increasingly we're moving from the idea of clinical syndromes to particular treatable traits. And I think we're going to start to see overlap in terms of what we might see in some trauma patients with what we see in infection and after surgery, and other mechanisms that will be very specific. So lots of people are involved in the COMBAT project, and similarly, I've spoken about a number of colleagues along the way who've contributed more widely. Thank you very much.
Thank you, Adrian. Are there any questions for Adrian? Adrian? I saw Adrian up there. Sorry, Julian. This one here.
Thank you. That was a great talk. I'm really interested in the differences and similarities between what you're seeing in different diseases.
Yeah.
Because my impression is that it's quite disease specific. Many host genomic findings are very disease specific in specific contexts. So are there, I mean, you've indicated this, but do you think there are opportunities to try and find some more generalized patterns? Because as you say, there are patients with flu, patients with pneumonia, patients with sepsis could end up in hospital with a very similar presentation, but have a very different underlying etiology, and therefore what you're actually treating. Sorry, I'm not a clinician, so I'm probably messing up the terminology, but what you're actually treating, it might not quite be the same. So is this a way to try and get through those challenges?
We hope so. And I certainly wouldn't want to imply that everything applies, all of these mechanisms are applying across different diseases, but I think some undoubtedly will be shared, and in some they could well be triggered by exposure of danger molecular patterns in the same way as if it's a pathogen associated. But the challenge is having the data sets to really start to explore that, and some standardization of approach that allows people to do that. And of course these will change over time, and so these sorts of assessments and biomarkers are probably going to have to be done through the patient's journey. And I didn't have time to talk about it, but if you look in that poor outcome group of sepsis patients, 3 months later, their neutrophils are still not normal. And if you put them with healthy T cells, they'll affect those T cells. And almost certainly that's epigenetically driven. So I think we're scratching the surface of the complexity of this, but what we're going to learn across what we currently regard as distinct diseases or syndromes is going to be exciting.
So a little bit of a follow-on there. So there are opportunities there with the different signatures there that you have in terms of point-of-care devices. So are you working with any sort of technology providers, looking at how you might put that onto a platform?
Yes, so as I say, with Cepheid, who specialise really in pathogen detection, for example, they have a COVID test, but they have the technology where blood can be loaded into a cartridge for a multiplex RT-PCR with a rapid turnaround. And if that can be then put into a machine learning framework, it's got the opportunity. But I suspect we're going to end up with a whole series of these types of tests as we increasingly understand the opportunities for stratification.
Yeah. Are there any other questions? This one there.
Thank you very much. I'm wondering, you've got great data there about people that have severe disease and predicting. Are you able at all to predict the people that are going to do well? Because actually, I think we all see people that do remarkably well despite their inflammatory markers and things. And having some indication they're going to do well may give them more confidence in early discharge and NHS pressures and things might support.
I think we don't understand that properly, but I think you're quite right. That's almost as important in terms of the clinical pathway, in terms of the level of monitoring and the sort of environment those patients can be managed in, including at home. So identifying those patients hasn't been a focus to date, but I completely agree. And there may well be genetic factors also driving that, because there are people who get repeated challenge for other reasons and do remarkably well.
Looks like it. Thank you very much again, Julian, and thank you for both of the speakers. And finally, over to Munir for some closing comments.
Okay, I need to close the conference. When I set this network up many years ago, I said that what we wanted was fantastic science, exceptional speakers, and a wide variety of areas to be covered. And I think today's been no exception. We've had a fantastic set of speakers and many sort of new areas that we've learned about today. So I just want you to thank all the speakers for the great talks today, please. I have to say that I've learned a lot today, a lot of new things, but the thing that sticks in my mind is the knowledge graph on Game of Thrones. I'll just have to study that slide. obviously, to organize a conference like this takes a lot of effort, and I want to thank the steering committee that helps with the UK Pharmacogenetics Stratified Medicine Network, and Karen, Anne, etc., and Laura, thank you very much. It's great that you were able to turn up today and so on. But also want to thank the British Pharmacological Society. The team are here. Too many of them are here for me to be able to name them individually. But thank you very much to all the BPS team. We've moved from Christine McNamee to the BPS team, but you know, you can see that the network is performing in the same way, retaining its same brand and so on. So thank you very much to the BPS team and to the RCP team as well for the AV. So thank you very much, guys, as well. And finally, thank you to you. You know, so you've been supporting, many of you have been supporting this network for many, many years, and you continue to support it, and we hope you can support it next year as well. And so please do come back next year for our annual meeting again, as well as our workshops, and I wish you safe travels home as well for today. So thank you very much, everybody.